So for our presentation, we received protein Argonaut 9. This is commonly known as AGO9, and this PowerPoint was produced by the following. So our protein is from the Argonaut family. These proteins were first discovered through genetic experiments of the cells and DNA, and through extensive research in the past couple of years, they have revealed lots of information about being the key players in gene splicing pathways guided by small RNA molecules and strands. So the Argonaut protein family was first discovered in plants. This was in the cells of the plant walls, and they were generally used for structure. Later, the same protein was found in common mammals, such as the house mouse, inside of their livers, and then furthermore, even into various types of fish, primarily studied in the skin cells of the zebrafish. Common biological processes of our particular Argonaut protein were defense response in the plants, which occurred in the same cells, gene splicing mediated via RNA, negative regulation of transposition, and finally regulation in transcription, and that was of course DNA templated.
What is Argonaut 9? Argonaut 9 is a component of the RNA into silencing complex, which is used in a process called RNA interference. The main purpose of RNA interference is to inhibit gene expression by cleaving and altering transcribed strands of mRNA. So in RNA interference, the first step is the cleaving of pieces of double-stranded RNA, either dsRNA or pre-miRNA, by using an enzyme called dicer. The resulting segments have a passenger strand and a guide strand. The passenger strand degrades, and then the guide strand becomes part of the RNA into silencing complex. The purpose of the guide strand is to recognize specific sequences in mRNA templates that are in the cell. Once the guide strand recognizes the mRNA sequences and the silencing complex binds to them, the Argonaut proteins, including Argonaut 9, cleave the mRNA strand and prevent translation of the protein from the mRNA, and they also prevent expression of the gene. It is through this mechanism that Argonaut 9 helps silence genes through RNA interference.
siRNA, or small interference RNA, is a synthetic RNA duplex designed specifically to target a particular mRNA. A 21 to 23 nucleotide RNA is processed from a double-stranded RNA by an RNase enzyme, and this interacts with the siRNA. This is required to silence periocentric located TEs in female gametes and their accessory cells. This means that it is situated near or on both sides of the centromere of a chromosome. This siRNA binding is necessary to deactivate a large proportion of LTRs. This helps to specify the cell fate in the ovule.
The Argonaut protein family is important in cell specification and gene silencing. This is done via siRNA binding, where siRNAs bind to specific mRNA targets and help to regulate gene expression. It is also done through interaction with and destabilization of the target RNA. In the common fruit fly, it is important in germline stem cell division. In germline stem cell division, germline stem cells can divide asymmetrically to form specialized cells. This is especially important in the formation of the female gamete. In cases of dysfunctional Argonaut 9, mutations in the Argonaut 9 protein can lead to defects in cell specification during megasporogenesis. This is the process by which a diploid cell undergoes meiosis to produce 4 haploid cells, or gametes. Another case where Argonaut 9 can become dysfunctional is when it is targeted by the turnip yellows virus. This can degrade the proteasome and disactivate the function in RNA silencing.
RNAi is likely to have the greatest impact as a therapeutic tool in 2 key clinical areas. These areas are cancer and infectious disease. The RNAi also has the potential to be a therapy for other disorders like dominant genetic disorders. In a recent study using mice, researchers used RNAi to knock down the gene TH, which encodes the dopamine synthesis enzyme tyrosine hydroxylase, which is important for normal functioning of the nervous system. By knocking this gene down, it resulted in behavioral changes and demonstrated how RNAi induced by RNA in viral vectors could be readily accomplished for specific uses, potentially including human gene therapy.
In several other recent studies, siRNAs have been shown to protect mice from viral infection, tumor growth, hepatitis, sepsis, and ocular neovascularization, a proliferation of abnormal blood vessels beneath the retina. There is possible application for human response in this as well. The siRNA approach for gene silencing holds great therapeutic promise, as siRNAs, like miRNAs, are naturally used by cells to regulate gene expression and are therefore non-toxic and highly effective. Potential uses. Why you should give us funding. Since the Argonaut proteins and the RNA-induced silencing complex are used to silence specific mRNA sequences, it is possible that they could be used to silence undesired foreign RNA sequences in the cell, such as RNA from viruses. This could potentially help prevent viral diseases. Possible medical uses. In the future, once more information is obtained regarding the silencing protein, it has potential uses of silencing common diseases such as various cancers. Although Argonaut 9 is not particularly found in humans, if modified to resemble the family's proteins 1 through 4, AGO9 could produce very promising results in the field of cancer silencing and prevention. Small silencing RNA strands could be one of the keys to eliminating one of the Earth's most feared diseases. Fund us and save our world. Shall we do that or no?
It's up to you.
I definitely had a couple stumbles, but it might be better to just delete it.